This research paper introduces a nonparametric Bayesian approach to reconstructing network structures and estimating dynamical parameters from time series data. It models the network structure A as a primary target, focusing on the marginal posterior distribution obtained by integrating over contagion vectors C, recovery rates gamma, and densities ρ. The posterior distribution is written in closed form, allowing for efficient sampling using a Markov chain Monte Carlo (MCMC) algorithm. The authors demonstrate their approach using the Zachary's Karate Club network and synthetic time series data generated with both simple and threshold contagion models. They find that imperfect reconstruction of the contagion function is possible with short time series, and that network reconstruction quality improves with increasing amounts of data, eventually reaching a plateau. In order to determine whether complex or simple contagion models lead to an easier network reconstruction problem, the authors perform a comprehensive simulation experiment using generative network models that capture structural properties observed in empirical networks, including the Erderani model, the network configuration model, and the clustered network model. The results show that there are structural and dynamical regimes where different types of contagions outperform each other. Simple contagions outperform complex contagions in regions corresponding to low basic reproduction numbers R0, while complex contagions outperform simple contagions in regions with high R0 values. The authors also find that complex contagions can accurately recover the number of links, or network density, even when they failed to place links correctly. Moreover, the authors observe that the performance differences between complex and simple contagions do not correspond to the differences in estimating network density. They identify regions where complex contagions vastly outperform simple contagions in estimating network density, which are caused by bimodality in the distribution of estimated ρ values inferred from simple contagion time series. Lastly, the authors analyze the reconstruction performance for nodes of various coreness and find that complex contagions outperform simple contagions for intermediate amounts of infection data, particularly for nodes with higher coreness. In the realm of network science, a significant challenge lies in inferring network structure from binary state data. This task is often complicated by the complexity of the underlying contagion process, which can be either simple or complex. A simple contagion is characterized by an individual's likelihood of adopting a behavior or contracting a disease being independent of the number of infected neighbors, while a complex contagion requires a certain threshold of infected neighbors to trigger the same behavior or infection. The paper presents a nonparametric approach to the network inference problem, focusing on the comparative analysis of simple and complex contagions. By utilizing a probabilistic model that does not rely on specific assumptions about the network or the contagion process, the authors demonstrate that complex contagions can outperform simple contagions in resolving dense networks, especially when the dynamics are intense. This suggests that different types of contagions may be better suited for resolving different network features, depending on their generative mechanisms. The study also highlights the importance of considering the one-core nodes in network reconstruction, as these nodes are less susceptible to complex contagion spread. Furthermore, the authors emphasize the need to categorize different types of contagions based on their ability to resolve local network features, rather than just global accuracy. This supplemental material explores the performance of simple and complex contagion models in various network models, including the small world network model and the stochastic block model, SBM. The small world network model is used to study the effect of small worldness and the clustering coefficient, while the SBM examines community structure. In Figure S1, we compare the performance of simple and complex contagion, where complex contagion is a threshold model with tau equals 3. The results mirror those seen in Figure 2, with the exception of the AUROC for the CM. This discrepancy can be attributed to the minimum degree of the power law degree sequence being 2. Making the network a perturbation of a two-regular network that is insufficiently connected to sustain a contagion requiring 3 infected neighbors to infect a node. Figure S2 illustrates the AUROC of the CIS model, the threshold model with tau equals 2, and the threshold model with tau equals 3 over their dynamical and structural parameters. This figure supports our main findings that R0 is sufficient to explain when simple contagion outperforms complex contagion. For instance, for the SBM, The epidemic threshold remains constant in the mean field limit for a fixed mean degree, and our network reconstruction appears to be independent of the structural parameter, E. The results in this figure demonstrate that our key takeaways hold, such as the epidemic threshold remaining constant in the mean field limit for a fixed mean degree, and our network reconstruction being independent of the structural parameter, E, for the SBM.